Welcome to Ælementz, your daily source of insights across finance, science, history, technology, and beyond. Whether you're traveling, tidying, exercising, taking a lunchtime stroll, or simply relaxing at home, we're committed to providing new content every day. So join us to grow and learn while living your busy life. Today, we're going to delve into CRISPR, a groundbreaking technology that's revolutionizing genetics and could transform medicine, agriculture, and many other fields in ways we never thought possible. What is CRISPR? CRISPR, pronounced CRISPR, stands for Clustered Regularly Interspaced Short Palindromic Repeats. While that might sound complex, at its heart, CRISPR is a natural defense mechanism found in bacteria. Think of it as an immune system for these tiny organisms. When viruses attack bacteria, CRISPR allows the bacteria to remember these invaders by storing a fragment of the virus's DNA. If the same virus tries to attack again, the bacteria can recognize and destroy it more effectively. Scientists have harnessed this natural process to create a powerful tool for editing genes. By guiding specialized proteins to specific parts of an organism's DNA, CRISPR can precisely cut and modify genes. This cut and paste ability means we can now change the genetic code of living organisms with unprecedented accuracy and ease. For example, if there's a faulty gene causing a disease, CRISPR offers the potential to correct that gene, effectively treating the condition at its source. The history of CRISPR. The journey of CRISPR began in the late 1980s when scientists first noticed unusual repeating sequences in the DNA of bacteria. These sequences were mysterious and didn't fit with what was known about bacterial genetics at the time. It wasn't until 2005 that researchers realized these repeats were part of an ancient immune system that bacteria used to fight off viruses. This discovery was like finding a key to a lock that scientists didn't even know existed. In 2012, two scientists, Jennifer Doudna from the University of California, Berkeley, and Emmanuel Charpentier from Umeå University in Sweden, made a breakthrough. They demonstrated how the CRISPR system could be adapted to edit genes in a lab setting. By simplifying the components and showing that CRISPR could target any gene in any organism, they opened the door to a new era of genetic engineering. Their pioneering work was so significant that they were awarded the Nobel Prize in Chemistry in 2020. Since then, CRISPR has rapidly advanced. Laboratories around the world have adopted this technology, using it to study diseases, develop new treatments, and even explore solutions to environmental challenges. The speed at which CRISPR has moved from a scientific discovery to practical applications is astonishing, reflecting its immense potential and versatility. How is CRISPR being used in medicine? CRISPR holds great promise for treating a wide range of genetic diseases by directly correcting the underlying genetic mutations. For instance, in 2019, a patient named Victoria Gray became one of the first people in the United States to be treated with CRISPR for sickle cell disease. This hereditary condition causes severe pain and organ damage due to misshapen red blood cells. Doctors use CRISPR to edit her own stem cells, correcting the faulty gene responsible for the disease. Since the treatment, Victoria has reported a significant reduction in symptoms, offering hope to millions affected by similar conditions. Another exciting application is in the fight against cancer. Researchers are exploring how CRISPR can modify immune cells to better recognize and attack cancer cells. In China, a patient with aggressive lung cancer received immune cells edited with CRISPR to target the tumor. While still in early stages, such trials could pave the way for new personalized cancer therapies. Can CRISPR help in agriculture? Yes, CRISPR is revolutionizing agriculture by enabling the development of crops that are more nutritious, resilient, and sustainable. For example, scientists have used CRISPR to create rice varieties that are resistant to bacterial blight, a disease that significantly reduces yields in many parts of Asia. By precisely editing the genes that make rice susceptible to the disease, farmers can grow healthier crops without relying heavily on chemical pesticides. In another case, researchers have edited tomato plants to produce fruits that ripen more slowly, reducing spoilage and waste. This has practical implications for food supply chains, especially in regions where access to fresh produce is limited. By improving shelf life, CRISPR-edited crops can help ensure that more people have access to nutritious food. What are the ethical concerns surrounding CRISPR? While CRISPR offers incredible potential, it also raises important ethical questions. One major concern is the possibility of making heritable changes to human embryos, which would pass the edits on to future generations. This was brought to the forefront in 2018 when a Chinese scientist announced that he had used CRISPR to edit the genes of twin girls to make them resistant to HIV. The global scientific community widely condemned this action as irresponsible and unethical, leading to calls for stricter regulations. There are worries about unintended consequences, such as off-target effects where CRISPR edits the wrong gene, potentially causing new health issues. There's also the fear of widening social inequalities if gene editing becomes a luxury available only to the wealthy, leading to a society where certain genetic traits are valued over others. These concerns highlight the need for careful consideration and international dialogue to establish guidelines that ensure CRISPR is used responsibly and ethically. Is CRISPR safe and accurate? CRISPR is a powerful tool, but it's not perfect. One challenge is ensuring that the gene edits are precise and don't affect other parts of the genome. Off-target effects can lead to unintended mutations which might cause health problems. Scientists are actively working to improve the accuracy of CRISPR. For example, newer versions like prime editing offer more precise control, allowing for single-letter changes in the DNA without making cuts in the double helix. Another aspect of safety is the body's potential immune response to the components used in CRISPR therapies. Researchers are investigating ways to minimize these risks, such as using delivery methods that target specific cells or tissues. Clinical trials are essential for testing the safety and efficacy of CRISPR-based treatments. By proceeding cautiously and under strict regulatory oversight, the scientific community aims to ensure that the benefits of CRISPR outweigh the risks. CRISPR could help eradicate malaria. Malaria is a devastating disease transmitted by mosquitoes, affecting millions of people worldwide. Scientists are using CRISPR to develop gene drives that can spread a genetic modification through mosquito populations, rendering them unable to carry the malaria parasite. In laboratory studies, this approach has successfully reduced malaria transmission. If implemented carefully, it could save countless lives, although it also raises ecological and ethical questions that must be addressed. Bacteria's ancient defense becomes our cutting-edge tool. It's fascinating to realize that a defense mechanism evolved by bacteria billions of years ago is now at the forefront of modern science. Bacteria use CRISPR to remember viruses that have attacked them, storing snippets of the viral DNA in their own genome. This allows them to recognize and destroy the virus if it attacks again. By uncovering this natural process, we've gained a versatile tool that could help solve some of humanity's most pressing problems. CRISPR and the revival of extinct species. The idea of bringing back extinct species, like the woolly mammoth, has moved from science fiction to scientific discussion, partly thanks to CRISPR. Researchers are exploring the possibility of editing the genes of closely related species to recreate traits of extinct animals. For example, by editing the DNA of Asian elephants to include characteristics of the woolly mammoth, scientists hope to reintroduce these creatures to the Arctic tundra, which could have positive effects on the environment. While still theoretical, it raises intriguing possibilities about conservation and ecological restoration. CRISPR is more than just a scientific advancement. It's a tool that could reshape our world in profound ways. From curing diseases and improving food security to potentially reviving extinct species, the applications are vast and varied. However, with such power comes the responsibility to consider the ethical implications carefully. What are your thoughts on CRISPR? Do you believe its benefits outweigh the risks? How do you think it should be regulated? Share your thoughts in the comments. Thanks for being here. If you find this helpful, consider subscribing. More content is on the way, and I hope to see you again.